Goldman-Rakic, who spent most of her career studying working memory and the prefrontal cortex, speculated that the capacity to hold information online evolved when neuronal firing persisted after the termination of a sensory stimulus. In other words, if a neuron continued to fire after an organism encountered a specific stimulus, a representation of that stimulus remained in working memory. This would provide a number of evolutionary advantages, such as the ability to track prey or predators as they move in and out of hiding. Natural selection, therefore, may have played an important role in the evolution of working memory. Of all brain regions, the prefrontal cortex has undergone the greatest expansion across human evolution. Measured as a percentage of brain volume, the prefrontal cortex is larger in great apes, humans, chimpanzees, gorillas, and orangutans than in any other animal. A primary difference between the brains of these close biological cousins is complexity. Humans have much denser connections between prefrontal cortex neurons and other brain regions, allowing more efficient communication between neural sites that control cognitive processes. It seems probable, therefore, that working memory was augmented over human evolution. It is also likely that the process was shaped by natural selection, because there is wide inter-individual variability in working memory abilities, which may have a genetic basis. Interestingly, performance on working memory tasks correlates with standard intelligence tests, but is influenced less by culture and previous learning than traditional measures such as the IQ test. Based on these two lines of evidence, prefrontal cortex expansion and heritability of working memory, some theorists argue that an increase in working memory capacity across evolution represents a qualitative shift in human cognition. Put simply, working memory allows individuals to think about something that is not there. This symbolic representation provides a mental blackboard for constructing solutions to environmental problems. Designing tools and using language are prime examples of cognitive processes that rely on maintaining and updating information in the head. This description can be extended to artistic works. The earliest known cave paintings were created approximately 30,000 years ago by artists who must have been imagining animals in their natural environment. These renditions could represent the beginnings of human culture, which began to flourish around the same period. Finally, because working memory involves the mental juggling of past and present events, it may form the basis of future planning, an enormous step in cognitive evolution. Although not everyone agrees, human enhancement in working memory capacity may be a final piece in the evolution of modern cognition. Reference memory. Just as discussions of short-term memory have been replaced by working memory, long-term memory has now been replaced by the term reference memory. Reference memory is a more accurate description of this type of memory because it implies an active process of referring to information in long-term store. Many researchers simply say memory to distinguish reference memory from working memory, but this text will employ the more specific use of both terms. The study of reference memory changed dramatically in the 1950s with a very unassuming gentleman who came to be known around the world by only his initials, Box 3.4. Box 3.4, an unforgettable amnesic. He knew his name, that much he could remember. He knew that his father came from Louisiana and his mother from Ireland. He knew about the 1929 stock market crash and World War II and life in the 1940s, but he could remember almost nothing after that. In 1953, he underwent an experimental operation to reduce brain seizures, only to emerge fundamentally and irreparably changed. He developed profound amnesia, losing the ability to form new memories. For the next 55 years, each time he met a friend, each time he ate a meal, each time he walked in the woods, it was as if for the first time. He could navigate through a day attending to mundane details, fixing a lunch, making his bed, by drawing on what he could remember from his first 27 years, but he could not hold a job and lived, more so than any mystic, in the moment. For those 5 decades, he was recognized as the most important patient in the history of brain science. As a participant in hundreds of studies, he helped scientists understand the biology of memory, as well as the fragile nature of human identity. In the 1950s, many scientists believed that memory was distributed throughout the brain and not dependent on any one region. That began to change in 1962 when Brenda Milner presented a landmark study showing that a part of the patient's memory was fully intact. In a series of trials, she asked him to trace a line between 2 outlines of a 5-point star, one inside the other, while watching his hand and the star in a mirror. The task is difficult for anyone to master. Every time he performed the task, it struck him as an entirely new experience, yet with practice he became proficient. At one point he said, "This was easier than I thought it would be." The implications were enormous. Scientists realized that there were at least 2 brain systems for creating memories, one for conscious and one for subconscious memories. This explains why people can ride a bike or play guitar after years of not touching either. He was a very gracious man, very patient, always willing to try these tasks I would give him, Dr. Milner reported, and yet every time I walked in the room, it was like we'd never met. Nonetheless, he was as self-conscious as anyone. Once, a researcher visiting with Dr. Milner remarked how interesting a case this patient was. "He was standing right there," Dr. Milner said, and he kind of colored, blushed, and mumbled how he didn't think he was that interesting, and moved away. On December 2, 2008, Henry Gustav Molaison, known worldwide only as HM to protect his privacy, died of respiratory failure in Connecticut, USA. He was 82. Hours after his death, scientists worked through the night taking exhaustive scans of his brain, data that will help tease apart precisely which areas of his brain were still intact and which were damaged, and how this pattern related to his memory. His brain has been preserved for future study, in the same spirit as Einstein's, as an irreplaceable artifact of scientific history. Henry Molaison left no survivors, but a legacy in science that cannot be erased. An important point about HM is that his intellect, as well as his language and perceptual skills, remained intact following his surgery. This helped to show that reference memory is independent of other cognitive processes. His ability to learn a new motor task without remembering having practiced it highlighted the distinction between knowing how and knowing that. Based on the thousands of research studies that HM's case inspired, this distinction has been refined so that reference memory is now organized into 2 broad categories, declarative and non-declarative memory, each with its own subdivisions. These are defined by the type of information that is processed and by a specific brain region that mediates this processing. The processing occurs via the 3 stages described in Section 3.2. There is no reason to believe that encoding and consolidation differ across the subclassifications of reference memory, whereas retrieval of declarative versus non-declarative memories may be distinct. Intuitively, recalling what you had for dinner 2 weeks ago is different from remembering how to ride a bicycle. The former, but not the latter, involves an awareness of searching for information in reference memory. For this reason, many people think that declarative memory is unique to humans, although, not surprisingly, not everyone agrees. Non-declarative memory. A number of clinical case studies followed HM, confirming that motor learning is preserved in amnesic patients. These studies also revealed other intact memories that were not measured in motor tasks, such as mirror drawing. Non-declarative memory was introduced as an umbrella term to describe these different types of memory that do not depend on awareness or explicit knowledge to be expressed. Habituation and sensitization. The simplest type of non-declarative memory is modification of a response based on prior experience with a single stimulus. The response may decrease or increase with experience, changes described as habituation and sensitization, respectively. Habituation develops to stimuli with low motivational significance. Sudden movement in the grass may elicit a startle response, which then declines when it is not followed by the emergence of a predator or anything else worth paying attention to. Habituation allows organisms to filter out irrelevant information, so they do not waste time or energy responding to a stimulus each time it is encountered. The specificity of the phenomenon provides compelling evidence that animals remember the stimulus previously encountered. This principle is illustrated in Figure 3.10. Habituation of aggressive responses in stickleback fish is most pronounced when the same intruder is encountered in the same environment. Figure 3.10. Habituation in stickleback fish. A. Male fish living in one environment were exposed to an intruder that was introduced in one of two locations in the aquarium. Aggression of the resident fish toward the intruder declined across a 30-minute session. After a 15-minute break, aggression of the resident was tested in 4 different conditions: the same intruder introduced at the same location, the same intruder introduced at a different location, a different intruder introduced at the same location, or a different intruder introduced at a different location. B. Aggression, measured as bites per 2-minute period, was lowest in the same condition and highest in the different condition. Okay, so A, you can see a diagram of the fish tank. So you have a resident on the bottom right of the fish tank, and then an intruder is coming in on the left. Okay, and then B, you have a graph. On the x-axis is time from 0 to 15, and on the y-axis is bites per 2 minutes from 0 to 160. Okay, so there's a bunch of lines that they're comparing, which are different fish, different location, same fish, different location, different fish, same location, and same fish, different location. So the most bites per 2 minutes is happening when it's a different fish in a different location, and the least bites are happening with the same fish in the same location. So they really are getting used to the intruder being in one area of the tank, and they're not too fussed by it. Box 3.5, Habituation Paradigm in Developmental Psychology. Young children love new toys, but often become bored with the same old thing. Parents take advantage of this by pulling long-forgotten items from the bottom of a toy chest. Scientists capitalize on the same effect using the habituation paradigm, which compares the amount of time infants spend looking at familiar versus novel objects. Figure 3.11. In a typical setup, a large visual stimulus, such as a striped pattern, is repeatedly placed in front of an infant. For the first few minutes, subjects spend most of their time looking at the pattern, but eventually ignore the stimulus and look elsewhere. At this point, the stimulus is removed and replaced with 2 stimuli. One is the previously viewed stimulus, and one is new. Babies, chicks, and infant chimpanzees spend more time looking at the novel stimulus, revealing visual habituation to the familiar image. Figure 3.11. Habituation Paradigm in Developmental Psychology. Infants view a visual stimulus presented on a computer screen. As time progresses, infants lose interest in the stimulus and spend less time looking directly at the stimulus. When a new stimulus is presented, final frame, interest is rekindled and infants spend most of their time looking directly at the stimulus. The habituation paradigm has been refined over the last 50 years, incorporating different stimuli across a range of ages. Indeed, hundreds of experiments in developmental psychology are conducted each year using a modified version of this original paradigm. These studies do not focus on habituation per se, but on the cognitive capacities of infants and young children. A primary goal in this research is to identify which cognitive abilities are present early in development and how these continue to develop in childhood. An intriguing example is provided by a study examining what infants understand about the physical world. Spelk et al., 1995. More specifically, do infants have a basic understanding of Newtonian principles such as continuity? Continuity describes how objects travel in a continuous path over space and time, with no gaps in either. In this study, infants were habituated to an object moving across a stage with 2 occluding screens. One group saw the object move behind the first screen. then reappear from behind the second screen without passing through the space separating the two screens. These infants subsequently took longer at a test display of one object than a display of two identical objects, implying that the former was more novel than the latter. The authors proposed that the infants recognized discontinuity of motion in the habituation phase. Since a single object cannot suddenly appear in another place, they inferred that there were two objects. Consistent with this interpretation, infants in a second group who were habituated to an object moving in a continuous manner, behind one screen, through the intermediary space, then behind the second screen and out again, looked longer at the test display of two subjects. The authors concluded that a cognitive understanding of object continuity develops within the first year of life. The debate about when these abilities emerge and whether any individual test is a true measure of this cognitive ability is ongoing. Nonetheless, there is little, if any, disagreement that habituation is a powerful measure of memory for a previously experienced event. In contrast to habituation, sensitization develops to motivationally significant stimuli or events that an organism should pay attention to. Sensitization is studied less frequently than habituation for the practical reason that increases in a behavioral response may be more difficult to detect, i.e., if the response is already at a maximum. Moreover, depending on the paradigm, repeated presentation of a potentially harmful stimulus could be unethical. Recently, the natural experiment of warfare has provided insight into sensitization in humans. Loud and sudden noises elicit a startle response that can be measured through electrodes placed around the eye muscles. War veterans suffering from post-traumatic stress disorder, PTSD, show exaggerated startle responses in the lab, suggesting that they are sensitized to noise bursts that mimic gunfire. Morgan et al., 1996. Interestingly, startle responses of veterans not diagnosed with PTSD are similar to a control group. Although there may have been pre-existing group differences in startle responses, the most likely explanation for the findings is that sensitization of a fear response emerges in individuals who are susceptible to stress-induced physiological changes. Current research is focusing on understanding the factors that predict this difference and whether it can be used to identify later mental health problems in high-stress occupations. Given the evolutionary value of filtering out or paying attention to stimuli that recur in the environment, it is not surprising that both habituation and sensitization are ubiquitous throughout the animal kingdom. Both describe a change in a behavioral response, so it should always be possible to identify sensitization or habituation of what, e.g., escape or startle response. Habituation and sensitization are sometimes classified as non-associative because they do not depend on learning a relationship between two or more stimuli. This position has been challenged by evidence that both habituation and sensitization are reduced, sometimes eliminated, in a new environment. This suggests that animals form associations between an environmental context and the motivational significance of a stimulus. Movement in the grass may be trivial in one location, but deadly in another. Perceptual priming. Perceptual priming is the facilitated identification of a stimulus as a consequence of prior exposure to the stimulus. Tulving and Schachter, 1990. In a typical human priming experiment, participants are asked to read words as they are flashed on a screen. The rate of presentation is so fast that most people identify only a small proportion of the list. If words are repeated within a short interval, accuracy increases, even though individuals report that they do not remember seeing the word before. A different task requires participants to name familiar objects from fragments of line drawings. Initially, only a small portion of the image is presented, so the pictures are difficult to identify. The image becomes recognizable as the lines are gradually filled in. On later trials, participants are able to identify the image at an earlier stage. Chapter 2 described perceptual priming in animals. Birds are better at detecting prey in a visual search task if the item appeared in previous trials. Priming is also observed in a bird version of the picture fragment task. Perceptual priming is categorized as non-declarative memory because it functions primarily at an unconscious level. Even patients with extreme deficits in declarative memory, like HM, show intact perceptual priming. The phenomenon has been observed in all sensory systems, with the strongest effects occurring when the prime and identifying stimuli are in the same modality. That is, an auditory stimulus may prime a visual stimulus, e.g., hearing a word may facilitate later identification of the written word, but the effect is reduced compared to two auditory or two visual stimuli. Moreover, it is not clear whether this cross-modality enhancement is true perceptual priming. More likely, it reflects semantic priming, in which a mental representation of the word meaning primes its later identification. Semantic priming is difficult to assess in animals because we have little understanding of the meaning that nonverbal subjects attach to different stimuli. If it does occur, which is probable, semantic priming would confer an evolutionary advantage, much like perceptual priming, that is, enhanced recognition and identification of frequently occurring stimuli would facilitate a number of cognitive processes, including categorization, decision-making, and counting.